Imagine discovering that the key to understanding a complex disease lies hidden within the intricate dance of genetic changes over time. That's exactly what researchers have uncovered in their quest to decode multiple myeloma, a type of blood cancer. By examining how genomic dynamics evolve, they've revealed insights into how these changes shape the course of the disease. But here's where it gets controversial. Not all mutations are created equal, and some may hold more sway over outcomes than others. Let's dive deeper into this groundbreaking research and explore why timing truly matters when it comes to genomic events. The study leveraged extensive datasets, including whole genome sequencing, WGS data from the GMMG HD6 trial, which has been made available on the European Genome Phenome Archive, EGA. These datasets, spanning EGAD 5700,681, EGAD 5768, and EGAD 5763, provide critical resources for scientists worldwide. Similarly, other datasets like MSKCC WGS, EGAD 00001132, and RRMM WGS, EGAS 0000106538, EGAS 0001004363, EGAS 0001048005, EGAS 0001059773, further enrich our understanding of the genetic underpinnings of this condition. What makes this investigation particularly fascinating is the way it builds upon foundational studies. For instance, Mora et al., in their 2020 review published in JAMA Oncology, highlighted the shift from focusing solely on cancer burden to exploring cancer genomics in smoldering myeloma. This transition set the stage for today's findings, showing just how interconnected genetic alterations are with clinical trajectories. But what does this mean for patients? Researchers now know that certain genomic events, notably those involving chromosome 1q amplifications or deletions, can predict whether someone will experience early relapse or respond well to treatments such as lenalidomide, bortezomib, and dexamethasone. And this is the part most people miss. Even subtle differences in when these genetic anomalies occur can drastically alter prognosis. Take, for example, the role of APOBEC mutagenesis, discussed by Mora et al. In a 2018 paper in leukemia. Their work demonstrated that hyper-APOBEC activity correlates with poor survival rates. Yet until now, no one had fully teased apart its temporal contributions across different stages of myeloma progression. Could mitigating APOBEC-driven mutations become a future therapeutic target? It's a tantalizing possibility. Another intriguing aspect involves melphalan exposure, explored in a 2021 study also led by Mora. Melphalan, a common chemotherapy agent, leaves behind distinct mutational scars that serve as markers of treatment history. By analyzing these traces, scientists can reconstruct the timeline of genetic events with remarkable precision. However, not everyone agrees on the implications of these findings. Some argue that while identifying high-risk groups is valuable, we still lack actionable strategies tailored specifically to them. Others question whether current classification systems adequately capture the complexity of genomic subtypes. Should we rethink how we categorize patients based on evolving genetic profiles rather than static criteria? These debates underscore the importance of continued collaboration. Initiatives like the Myeloma Solutions Fund and partnerships with institutions such as Memorial Sloan Kettering Cancer Center and Heidelberg University play pivotal roles in advancing knowledge. Together, they ensure access to cutting-edge technologies and foster multidisciplinary approaches necessary to tackle challenges ahead. So, what do you think? Do you believe prioritizing dynamic genomic assessments over traditional staging methods could revolutionize care? Or should we focus instead on refining existing frameworks? Share your thoughts below. We'd love to hear your perspective.